Such an amazing conference, so thanks for having me, Steve and Matthew. And it's also kind of a great pleasure for me to be the warm-up act to such beautiful and exquisite minds that are going to follow me. So I think what I'm going to do is somewhat of a kaleidoscopic type of stream of consciousness intervention. We'll see how it goes, and then it will be up to you not to actually try to unpack what I'm going to do with you in the next 50 minutes or so. Also, after something froze here now. That's quite nice. Let me try again. I like technology. As you can see, the computer is now totally frozen. That's why we like to work with life, because when you lose control over life, you actually can blame someone else. So, anyway, after Georgia Gammon's talk the other night, I realized that actually maybe I'm not an artist, although literally what we're doing is working with art as a life forms, but the type of life forms that we're working with are somewhat different, I would imagine, than what Gammon was kind of referring to. And then I thought that maybe, rather than be a shoemaker, I'd rather be a gardener. So let's read the next 50 minutes in the context of gardening. And it also reminds me that the thing that actually started this whole project to a large extent, and that's reading a paper by Ezio Mazzini, the Italian design thinker, who was talking about a future in which the made environment would be more like a grown environment. So he was thinking about objects that are not instruments for our use, but rather entities that are effectively linked and need care. Think of objects as plants in our garden. Think of objects as that are beautiful and useful as trees in your own garden, objects that endure and have lives of their own, objects that perform services and require care. So he was thinking about this new criteria of objects, not necessarily really living objects, but I took it literally. Now, there's no better audience than yourself to realize that something might be very wrong with this picture. The idea of actually changing the culture from a culture of manufacturing to a culture of growing, talking about our made environment as if it's a garden, I think there might be some issues that we need to resolve in regard to that. And that's really what United myself was set up to do since 1996, to figure out what's wrong with this picture. And throughout the years, we were kind of trying to develop different types of ways to engage with what we've been experiencing. And one of the things we are coming up just now is this whole idea of Neolifism. So we talk about it in terms of the fact that now we have fragments of biological bodies, forms of lab-grown life, and they require a different type of epistemological and ontological understanding, and by extension, and as hopefully I'll demonstrate, a different taxonomy of life. The liminality of this kind of technological approach to life can lead to a form of fetishization, or fetishism, which we call Neolifism. So again, this is kind of just an establishing statement, and hopefully as we go through it, you'll understand what we're trying to say. So back in 1996, I approached the scientists with this idea of trying to explore the use of living tissue as a medium for artistic expression, to kind of try and unpack what Ezio Mazzini was talking in regard to creating this new made environment, which is like a garden. And the technology of choice that we decided to look at was what was called tissue engineering, which is really the birth of this whole field of regenerative medicine. And that scientist sent us to a colleague of her who was working in an eye research institute. He was trying to figure out if he can develop a new artificial cornea. And just keep that in mind, because the very first thing that we've seen when we arrived to this lab was this. Those are half rabbit's heads. And those rabbits have been, or so we've been told, killed for food, for a gourmet restaurant. And twice a week we would get this box with those half rabbit heads. The heads would be sent after being slaughtered to a brain research institute where the brains would be taken out, and around lunchtime you would receive the box with those rabbit heads, from which you would then pull out the eyes. And then, as you can see here, we would then put those eyes in antibiotic solution and leave them in the fridge overnight. And just do eyes. Because when we would look down the microscope a few days later, we would see that. The cells would be alive. So although we apparently and obviously seen dead rabbits, or not just even dead rabbits, just parts of dead rabbits, in the cardboard box. 24 hours later, suddenly, some part of those things were alive. And we couldn't even articulate to ourselves what's there, what are we dealing with. Because those cells were obviously alive, we would watch them growing and multiplying and doing all of those kind of things, but they were not alive like the rabbits, where they were alive. So we had to come up with our own term, which we refer to as semi-living. The idea that it's alive to some extent, but not alive to the full extent of liveness, if you like. And we developed a series of projects, and then in the year 2000, for the very first time, we were able to grow our first tissue-engineered sculptures within a cultural context. So before we would grow everything in the lab, in 2000 we were able to grow our very first public, if you like, public display of tissue-engineered sculptures, and they were semi-living worry dolls. Now, I'm not going to talk too much about this project. I'll just talk about something which is, again, really, really strange that happened. Because when we started to work with labs around the world, we would ask the lab to give us a list of the type of cells they were working with, because we didn't want to travel with living tissue. Crossing borders with living tissue beside your own tissue is a bit of a problem. So we would get a list, and this was the very first time, and we got a list from the Department of Virology of the local hospital in Linz, where we showed the work, and one of the cell lines that was on the list was a cell line called McCoy. Now, this is kind of the way in which cell lines appear within the tissue banks, within the American Tissue Culture Collection. This is kind of information one gets when you do a search in regard to different cell lines. And the cell of choice was McCoy. From my perspective, being a Trekkie, the idea of having the first tissue-engineered sculptures cells named after McCoy, the first Doctor of Star Trek, was kind of good enough. But what was even more interesting was this description that tells us that those cells originated from a human, from a fluid of an arthritis patient, but now they're classified as mouse cells. Somewhere along the line, those cells have been transformed, or obviously mislabeled, but those cells have been distributed around the world for many years as if they're human cells, while actually being mouse cells, the strain L mouse fibroblasts. And that started to kind of click that actually the world of tissue culture, the world of those fragmented parts of bodies that we're working with, falls outside any type of both cultural but even taxonomical and even biological way of referencing them, and it's a very, very messy and confusing world. So the only thing we can kind of refer to it is maybe something akin to the cabinet of curiosities. You know, what we were unpacking and opening up was this really strange cabinet of curiosities that only exist within this context of those research laboratories. And we try to kind of figure out again how we put that within a cultural context. So for that also we need to go back. So we need to look at kind of this idea of the industrialization and the creation of technological bodies in which those fragments or those parts of life operates. So this is what was called an artificial mother. This is the first chicken incubators and a way to keep chickens within kind of artificial environments to be able to get a constant supply of chickens throughout the seasons, especially within cold climates. So this is kind of one way to look at the source of what we are looking at. The other thing is this idea of the meat packing, which is actually the birth of the mass production and the industrialization. Story is that Henry Ford went to one of those meat packing or the disassembly lines, if you like, and that's where he picked up the idea of the assembly line. So the idea that life was what brought the assembly line. Fordism actually started by actually disassembling living things into the fragments, into the parts. Jumping all the way to 1895, we're starting to see a very different logic applied to life, which can be summarized by a misquote from A.G. Wells from a paper that he published, which titled On the Plasticity of Living Beings, where he talks about the fact that we overlook, which is no longer the case, but then they overlooked only too often the fact that a living being may be regarded as a raw material, as something plastic, something that may be shaped and altered. How many of you find that disturbing? No? Okay. It's a generational thing, because I use this quote for many years now, and less and less people actually find it disturbing. And when I ask them, so what will happen if we change living being with human being? And they would say, actually the young, I just gave a similar lecture to high school students. They said, that's okay, as long as someone else is doing it. And someone else thinks about it as well. Anyway, so this idea of life as a raw material already appears in 1895 with A.G. Wells, and to a large extent because of work of people like Jacques Laub, who already in the late 19th century, beginning of the 20th century, was thinking about the dream of biology moving from a descriptive discipline to a prescriptive discipline, biology moving to an engineering discipline as opposed to something which is merely analytical. Life is raw material, biology is engineering. And again, actually, yesterday when Rosie was talking about Marxism looking at nature as an obstacle to be overcome, this is also the logic of factory farming. And I would argue that our attitude towards factory farming would inform our attitude towards a new biology and the new way we're engaging with life. And this is one of the first industrial scale chicken incubators, and you can see the language of engineering and profit is the main language, the standard of the world, raising poultry for profit. The main person that I kind of interested in is this guy over here, Alexis Carrel, who won the Nobel Prize for Medicine in 1912 and is credited as the person who started the whole field of tissue culture. He is the person who actually recognized that you can fragment parts of bodies of complex organisms and maintain them alive within an artificial support for long durations of time. He was trying to find the key for immortality, and he actually had the biggest mouse eugenics program in the United States as well at the time, as he was doing that. But in order to maintain the life of fragments outside of the original body, you need to create a new kind of body in which to grow the cell, in which to grow those fragments. And this is what we refer to as the Techno-Scientific Body. And this body, combined with life, if it's not well culturally articulated, would take us to some very strange places. So this is one of the very first premature baby wards in the United States. This is in Buffalo, actually the same place where the Cypher incubator was developed. This is in New York. And the reason for those crowd control rails there is because it was in a freak show. People would pay money in a freak show. This is Coney Island. And you can see there it says infant incubators with living infants. The only place where body and technology could come together was in the context of the freak show. And it lasted for about 30 years. It had 80% success rate, as opposed to zero if those babies would be left alone, and it would still be only operated within the context of the freak show. 1929, A.G. Wells and Julian Huxley write a book called The Science of Life, where it starts with the sentence that starts the whole book is, "The body is a machine." And within that they have a chapter about tissue culture, where they talk about the idea that actually tissue culture is kind of a very strange thing, because if the cells are in the body and within the organs, so the organ, such as a brain or liver, is like the city during working hours. Tissue culture is like Regent Park on bank holiday, a spectacle of rather futile freedom. A freak, yeah? It's just what it's doing. But also, this is kind of the statement of the body as a nation state. Yeah? If the organ is a city, the body is then a nation state. And when you think about it in this context, you see the next project of Alexis Carrel, who collaborated with Charles Lindbergh, was to develop this artificial or an organ perfusion pump, or as it was referred to as a chamber of artificial life or an artificial heart. And in a sense, another piece of scientific technology which is designed for display. Here you can see actually Charles Lindbergh and Alexis Carrel, and Charles Lindbergh is kind of breathing life into this city which is isolated from the nation state, into this organ that is kept alive in this chamber. They published a book together in 1938 called The Organ of Cultures, The Culture of Organs, but also Carrel very shortly afterwards went to a lawyer saying, what is going to be my responsibility towards someone I'm resurrecting? Not less. Yeah? It's like, okay, I need some kind of legal advice to figure out where's the biopolitics here. But also he published a book called Men the Unknown, 1938, where he recommended the use of gas chambers to eliminate undesirable elements in human society. Those rogue cells that are floating around, living the nation state, should be economically disposed of within those gas chambers. Yeah. So the question is, what came first? His relationship to human society in such a way that made him think about the fact he can fragment a body, or vice versa. And I'll keep it open. 1948, we see, going back to our good friends, the strain L mouse cells. This is the first continuous cell line that was developed. So when you take cells directly from a body, usually they have a finite amount of division they can go through, but there are types of cells that can actually divide forever. And in the scientific world, they're referred to as immortal cells. Most of them we recognize as cancerous cells, and that's one of the issues. And this is a cancer cell from a mouse that was isolated in 1948, started from one cell and then continually grown and basically continuously living since that time. So it's more than 60 years, and it's basically the oldest piece of living mouse tissue in the history of the universe, if you want to think about it in those terms. If we would collect all of those cells now, they would fill up this building. Similarly, in 1951, the first human cell line, the HeLa cells, and I'm sure most of you would know about Henrietta Lacks cells, that her cells from a cervical cancer were isolated and are being used for research still today. And again, the biomass of those cells is way exceeding the biomass of Henrietta when she was alive. But I won't go too much into that. It's a very long story. 1954, the whole field of tissue culture finally is becoming standardized, mainly through the work of White here, who was also kind of very critical of the way Carrel was operating. And this is a really beautiful theatrical quote of the way the theater of the Carrel's tissue culture laboratory looked like. Very same year, interesting enough, there was also the first successful kidney transplant. So this idea of standardization of fragments of life was also to 1970s, another interesting thing came about, and that's what's called tissue or cell fusion. The possibility to be able to fuse cells from different organisms together and make them into viable cells that can still divide. This idea of the chimera actually comes to life, and you can see this beautiful quote. And the interesting thing is that Harris decided to declare a self-moratorium on his work when he realized that he can actually fuse a carrot and a frog cell and cross the boundaries of kingdoms within the living taxonomy. But anyway, those hybridomas, that's how those cells are being referred to, are still being used, and they are still being in operation and circulation today. And you can see this is again an American Tissue Culture Collection catalog, and it talks about this hybridoma, which is a mixture of mouse, a hamster, and yet another mouse. So 3 cells that were combined together. This is another one. This is Homo sapiens and a mouse together. There was very little outcry about that because there was something else there to deal with at the time, and that was in vitro fertilization that created so much sensation. So those things seem to coincide quite well. 1991, there was an attempt to try and make sense of it, and this is a paper that was published in the History of Evolution journal, where there was an attempt to make sense of the HeLa cells that have been growing, and basically saying here is a new type of microbe that needs to have a new designation. It's originated from a human source, but it's now a totally different type of organisms living within a specialized environment, specialized ecosystem, and that's the ecosystem of the laboratory. 1995, another thing which from our perspective was extremely important, and that's the appearance of the most visible chimera, and that's the mouse with the human ear growing from its back, and Yonat will talk a little bit more about that. But that's kind of what prompted us also to some extent to develop our project and to choose this technology of tissue engineering as a technology of choice in order to problematize this whole area. In the year 2000, we also had an exhibition which was titled The Stone Age of Biology, where we were scanning and miniaturizing those prehistoric stone tools and then growing muscle and nerve cells over them. And the idea was fairly simple. The idea was that the same way that our ancestors realized that they can chip away stone to create tools and by that transformed us into this technology-based organism that we are now, we're now starting to chip away life. And then we had a series of other works, including this piece over here, which was our first in vitro meat. So we developed a series of works which we titled the Technologically Mediated Victimless Utopia. And the idea there, and actually it was quite interesting to listen to Rosie yesterday, the idea there was to look at how Western technology is doing so well in hiding the victims of our consumption to such an extent that we don't really need to make any ethical decision when we're consuming the world around us. And in vitro meat is a great example of that. And soon, actually, in about 3 weeks, you're going to see the media would be splashed with a Dutch scientist called Mark Post, who is going to serve a hamburger-size in vitro meat to some celebrity in London, and be this really interesting rhetoric around it. And we'll be more happy to talk to you more about it. But in the meantime, I just want to show you kind of a video
Western Australia
that would explain it a bit more.
A strange creation is slowly growing in the lab. Oron Catts works in the Biological Sciences department. He's making some of the world's first artificial meat.
When we grow those cells, we actually tend to refer to them in terms semi-living, because they're not fully alive, they're not an animal, but they are a living part of an animal sustained in an artificial environment.
This artificial meat was grown from the muscle cells of a lamb.
Now note the size of the biopsy.
From any animal. The cells were cultured in a nutrient solution made up of blood plasma derived from cattle. The muscle cell cultures are grown in a microgravity bioreactor, which constantly rotates the meat so it grows in 3 dimensions. Without this motion, the meat would be flat, only a few cells thick. But there's another problem to overcome if the lab-grown meat is ever to mimic the real thing.
There is research which is being taken place at the moment in trying to find ways in which you can exercise those muscle cells so they would align and have more of a muscle architecture. And there's 2 ways that can be done. One is by electrically stimulating those cells, or the second way is to try and exercise them physically, trying to build tiny exercise machines, tiny gyms for those cells to grow and become more like muscle.
Oron has brought his unique piece of engineered meat to the UK.
Note the size of the
chefs. With 3 Michelin stars, Heston Blumenthal is famous for fusing science and gastronomy. This meat is so cutting edge that in Britain it's not recognized as food and so can't legally be eaten.
It's got a smell of raw liver to it.
I never really think it, but
You're going to turn it into the world's most expensive, tiniest hamburger.
Yes, which it is at the moment, and it's as big as one can get at this stage.
Many people may find the notion of lab-grown meat less than appetizing, but not this master chef.
I think as a catalyst for conversation, debate and thought, it's fantastic because how many people are quite happy to eat sliced ham that actually is not ham at all, it's processed meat from animals that have been literally chained to the floor.
Yeah.
That's not a problem, but something that's been grown in our lab actually is a problem. To me, that has the potential to be a lot more ethical.
The technology is still in its infancy. Even when scientists figure out how to ramp up production, this lab-grown substance would only work as ground meat. Engineered steaks and chicken breasts are only in the dream stage. And none of it is economically viable to produce, yet.
So just to point out, this was a BBC Discovery documentary that was dealing with extreme technological solutions to global warming. So basically, attempts of extreme technological solutions to solve problems that were created by technology in the first place. So this is the only reason, because it seemed to be more like an artistic intervention. It was aimed to be broadcast, and it was just before the Copenhagen Convention that was supposed to replace the Kyoto Convention, as a warning sign. So that's why the reason I was willing to be exploited in such a way for the promotion of such a crazy idea, because from our perspective the interest wasn't so much the meat itself, but it was this idea of the technologically mediated victimless utopia. As you remember, they mentioned that we still use fetal calf serum, so basically blood plasma from a cow. In order to grow this steak, we needed to actually use blood plasma from one calf. So actually a calf was killed in order to feed this tiny steak that was actually smaller than the original piece of the lamb that we picked up. So that's how absurd it is. So it's very similar to the way we all wage war at the moment, as we heard yesterday, but it's also very important to realize that this is that people like the people for ethical treatment of animals are recommending this type of solution as well. Because I would claim that quite a lot of our ethical decisions and the way those kind of things are being operated at the moment are much more on the aesthetic level than the real deep ethical engagement. Anyway, what we've seen as well, the video was the 3rd time we've grown a steak. The original steak was grown in Boston when we were residents in Harvard Medical School. We couldn't eat it then because the laboratory didn't have a license to grow food. But the 2nd time, which was part of a show in France in a place called La Lieu Unique, an exhibition called L'Art Biotech, we were able to build a fully functioning lab in the gallery with a dining room, and for 3 months we would go every day to the lab and feed our steak, and we were playing on this idea of what constitutes foul food. So we knew that the French people don't like the idea of eating engineered food, and we knew that most other people don't like the idea of eating frogs, so we combined those 2 things together and created our tissue engineered frog steaks, which And we also kind of saved 2 frogs from the local edible frog distributor. And in the last day of the exhibition, we had this ultimate nouvelle cuisine dinner where we served in vitro meat steak to some members of the audience and some people we invited, and released the 2 frogs to the ponds of the local botanical garden, only to be eaten by ducks shortly afterwards. But also, out of the 8 people that joined us for dinner, 4 of them spat out the steaks, which was a great thing for us because we could then collect them and show them in our follow-up exhibition called The Remains of Disembodied Cuisine, which was a video documentation plus the bits that were spat out. So, this is kind of the way we do our interventions. But now let me kind of just go to another area, and that's really kind of the bulk of the talk, and that's this idea of Neolifism. And in a sense, the steak is a great example of Neolifism to some extent. And again, I would recommend that you follow carefully what's going to happen in the next couple of weeks, because there'll be a prime example of media hype around Neolifism. But I can track the best example of Neolifism, obviously, to the Human Genome Project. And I don't know how many of you remember, in June 2000, there was a big ceremony, both on this side of the Atlantic and on the other side of the Atlantic. In America, they had Bill Clinton, Craig Venter, Collins, all celebrating the so-called completion of the first draft of the Human Genome Project. Yeah? And you see, decoding the Book of Life, a milestone for humanity. You have like amazing statements. So on this side of the Atlantic, the head of the Wellcome Trust said the following: a few months ago I compared the project to the invention of the wheel, but on reflection it's more than that. This code is the essence of mankind, and as long as human exists, it's going to be important and will be used. Interesting enough, the same people that have done that now saying that the Human Genome Project will be only remembered as a footnote in the history of science. And one of the reasons for that was that this whole ceremony in June 2000 was a political stunt. Now you remember in the very same spot, at the beginning of this conference, we had the geneticists saying, "Trust me, I'm an authority." Look what they're saying. While the timing of the announcement may have been directed more by political than scientific criteria, the joint announcement was probably more grandiose than the situation warranted. How do they want us to trust them? And what have they done? They created such a hype around the essence of mankind, which is this bunch of DNA, which is really kind of the only package that the human male is delivering, and therefore it's important and doesn't have any other context. That's really what they're kind of trying to make us believe. And what came out of the Human Genome Project? Diagnostic tools. No therapy, no cure. And actually to do with business plan, that's a much smarter business plan for pharmaceutical companies to create diagnostics rather than cures. So here we are. And then the next thing, so that once they realize it doesn't really work, the same people that are developed the Human Genome Project are now embarked on the Microbiome project. And that's trying to map all of the bacteria and other life forms that share the body with us. Because only 10% of the cells in our body actually constitute of human cells. 90% of the cells that constitute our body are actually considered to be foreign cells. And because those cells cannot be cultured in laboratory conditions, there was very little that was done with them until recently with the technology of metagenomics, which is actually developed by the very same people that did the Human Genome Project. And just wait for those next set of diagnostic tools that would diagnose the type of microbiome that we all share. Well, it will. Then just another area is kind of a really interesting thing, which is to do with context. This is a work of a Stanford scientist called Erwin Weizmann, where he actually went to the ethics committee of the state of California because he did research that proved that if he puts a human stem cell in a developing mouse embryo, the stem cell would develop into part of that mouse body, including in the brain. So he was already able to create a mouse, the 10% of the brain cells were human cells. And he says, what will happen if I create a mouse that would have 100% of the brain cells, human cells? And the response was, we'll have to stop it once the mouse starts to behave like a human. And how do you do it? Because the mouse is a model for the human already. You know, how much more you want to make it into a humanized mouse before you should stop. And then we're going to this idea of what really constitutes Neolifism, or the way we observe life. So this is kind of a traditional natural history museum. This is from Nantes in France, where the form of the animal, any of you who's involved in taxiderming animals knows that actually those things that you see in the museum are actually idealized form of that organism. The only thing which belongs to the original organism is the hide. Everything else is sculpted. It's actually an idealized form which is sculpted on which you stretch the hide of the animal. The only thing which is kind of still odd is how the human is presented within the context of the Natural History Museum. This is kind of still akin to the Cabinet of Curiosities, and I was really surprised to see that was in 2003, that they still had a Maori warrior head on display as an odd thing within this continuum of life. And a skin of a French soldier, a mummified, or a mummy, Egyptian mummy, and another skull over there. But the whole idea of the Natural History Museum and the collection and the whole idea of observation was really based on the form. But through Neolifism, things are changing. So the only thing that we worship now to do with the form are things like those, how we represent those new life forms. So this is the death mask of Dolly in the Natural History Museum in Vienna. This is a sculpture of our friend, the mouse with ear on its back. And if you really want to worship it, you can go to the Science Museum in London and worship Dolly's droppings, which are on a pedestal as well. But really, this is how natural history museums operate at the moment. This is how the collection looks like. This is from the Natural History Museum in Queensland, or in Brisbane, in Australia. And this is how the collection looks like. Boxes of frozen samples of tissue and DNA. Obviously, it's not very evocative, but what it stands for privileging this information, which is supposedly embedded within those tissues and DNA, as if that's standing for the animal. And to do with the information, Craig Venter, again a good friend from the Human Genome Project, in 2010 announced that he created the first self-replicating cell that we have on the planet that is apparent as a computer, which is, again, from a kind of a patriarchal point of view, it's the wet dream of every geek. Yeah? Because it's totally decontextualized. And what they've done there was actually very little to do with science. It was an extremely advanced technological fit, but what they've done was to photocopy, basically use a computer as a glorified photocopy to sequence a piece of DNA that they didn't even know the meaning of it, and then output it, and using actually other organisms like yeast to then sequence it again and insert it into an empty cell, a cell that the original DNA was removed from. But what I find also quite interesting is the fact that this is the image they used in order to kind of elicit response from the audience or from the public. This gaze. We heard yesterday about the fact that you only can objectify something by looking it into the eyes. That looks back at us. And this is the bacteria, the first bacteria that was one of the parents is a computer. Another example, which I think is actually even more telling, this is this place called Frozen Ark. It's a consortium of quite a few natural history museums and zoos around the world where they keep frozen samples of endangered species. It's like the seed bank that also we heard about yesterday. But if you go to their website now, this is kind of their Christmas special. And it's a perfect Christmas gift because for £250 you can sponsor the DNA of one of the animals. For £600 you can keep living cells which could allow future resurrection of the species. If you pay, £10,000 or £6,000, which is 10,000 Americans, this will allow the Frozen Ark project to survive forever. The concept of time is a bit strange there. But this idea that you can reserve, and actually you get a personalized PDF as well that kind of commemorates the fact that you sponsored that. Yeah, but this idea that this piece of DNA or cell that is sponsored is standing for the animal is quite strange. And when you think about it, and actually I'll come back to this later because I want to show you, this is kind of where it's coming from. It's coming from all of those different resurrection projects that are taking place. This is the Long Now Foundation. Some of you might know Stuart Brand, the guy who wrote the whole Earth catalogue in the 1970s, and he's now talking about this idea of the resurrection. And what he's saying here, that would mean a species is not completely extinct until we know there is no way to discover or deduce its full genome. Yeah? Forget about the cell in which this DNA needs to operate as a database. Forget about the embryo that this needs to develop into. Forget about the uterus in which it's being developed. Forget about the environment, the ecological system in which it operates. It's only the genome. Everything else can go to hell because we have the potential embedded within this information. Forget even about the microbiome that every organism has, which is specific to them. It's just the very small package that the male organism inserts and takes. It's the homunculus. Yeah? And let's see this. This is something I just received recently. This is a conference which is about to take place in April next year in Cambridge. to do with how will synthetic biology and conservation shape the future of nature. And that kind of rhetoric that's used is unbelievable. It says, in a sense, people have already mastered the planet and engineered it for their own uses. But more interesting, those discussions are based on a view, so discussion of conservation are based on the view of the natural world that does not include the re-engineered, resurrected, and novel life forms that are or soon will be created. What is the conservation community to do about genetically improved species, and so forth, and so forth. How messed up things are. You know, if this is Neolifism, which it is, we really need to figure out how we're dealing with that. And I don't think that this conference, which has bees with DNA growing off their wings, is really the best way to deal with it. But let's see. Anyway, so we did series of works around that, including kind of a natural history style display that starts with the two-headed birds that stands for the Cabinet of Curiosity, goes through this natural history display, and then ends up with a bioreactor that has hybridomas, those life forms that are falling outside of any type of classification, totally abstracted within this technological body. So when people come to see this work, they obviously can kind of identify with what's going on there, but the only thing which is alive is this abstracted life within the technological body. Okay, now I want to conclude. It's a little story that will take me about 10 minutes, hopefully, to tell you, but I think this is another really interesting story about what happens when one is being inflicted with Neolifism. So all of those examples I was showing to you were kind of to do with, the situation where our society is being inflicted with Neolifism. Now let me, and allow me to self-indulge and tell you my own story. So last year I was invited by the Bioart Society, the Finnish Bioart Society, to host a little group that would go up to the very far north of Finland to a place called Kilpisjärvi, which is the big kind of the border between Finland and Norway and Sweden, the very, very top north of Finland. And what I've done there was, with Jonat, we realized that we were exploring the idea of the semi-living, the living tissue. We kind of touched with our work also on the idea of the technological body, but there was something which was missing, and that was the substrate, the context in which all of those things operate, the way the substrate, the actual thing that appears in all of our works, but we never really explicitly discussed how that affects the life, how important that is. And it's a lot to do with quite a lot of research that we read talking about the importance of the matrix, the extracellular matrix on life form. So my idea, going to the very far north of Finland, was that I would reach a place where there will be only ice and water and rocks. And I hope that there I will be able to find something like a proto-substrate that I'll be able to use in order to kind of start this project and generate something that we can grow from the life that we were interested in. But to my surprise, and that was September last year, it was still full of life. It was teeming with life. And being based in Australia, I know that if I go to the bush after 2 days, I die. Over there, I could survive and actually sustain my biological body for a few weeks until winter would come, with berries and mushrooms and maybe hunt a reindeer. And there was stuff there that was so alive, and every step that I took was kind of just this layer and layer of life that I couldn't connect to. And I found it really, really strange, but I was trying to find this proto-substrate that was really hard for me to deal with, until one of the days we went on a field trip and we found this site. And that looked as if there was a fire there a few months earlier. And there were like mingled metal, and it was kind of a very strange place within this kind of totally pristine environment. And we came closer, and you can see obviously it looks as if it's quite a recent fire that took place there. And then we've seen this sign, and the sign tells us that in 1942, 70 years ago, 9 German Junker aircrafts formation was traveling, and one of them crashed there and exploded. So this is 70 years old crash site of a German plane, a Junker 88. And then suddenly it hit me. There were quite a few stories that I realized that are coming together with that. So this is how those Junkers look like. And one of the things I found there was this small piece of plastic, which I'll come back to shortly. But another thing I remembered was that a good friend, Charles Lindbergh, the one who created with Alexis Carrel the organ perfusion pump, was the first American to actually be permitted to see this very same type of plane, the Junker 88, because he was an advisor to the Luftwaffe. He was going initially was sent by the Americans, but became in love with kind of the German culture and the German Luftwaffe, And in 1939, he also published in Reader's Digest this essay trying to convince the Americans not to join the war because the war is against someone else. And basically, listen to what he's got to say. Aviation seems almost a gift from heaven to those Western nations who are already the leader of their era, strengthening their leadership, their confidence, their dominance over other people. It is a tool specifically shaped for Western hands. Yeah? A scientific art, which that's another reason why I shouldn't be an artist at the moment dealing with science, which others can only copy in a mediocre fashion. Another barrier between the teeming millions of Asia and the Grecian inheritance of Europe. One of those priceless possessions which permit the white race to live at all in a pressing sea of yellow, black, and brown. Think about the drones for a second that we heard about yesterday as well, and make this connection. So, okay, here's one thing. The other story about this small piece of plastic is that the birth of contemporary biomaterials, the same science that we were starting to explore when we entered the lab back in 1996 that was researching this artificial cornea, was to do with the fact that German and British pilots that crashed and got past part of those cockpits, this PMM, or as we know it as plexiglass, in their eyes. The eyes didn't reject them, and that was the birth of the development of synthetic biomaterials for implants. And in a sense, after a few generations, the same type of, or the lineage that led to the mouse with the human ear growing from its back, the same type, or the material that was used was kind of that lineage. And in a sense, the birth of this whole field of regenerative medicine, all started by this type of crest site. Yeah. So I had to go all the way to the very far north of Finland, to a place which was like the most remote place I ever been to this site of wilderness, only to connect with a small piece of plastic. And that was quite disturbing, and especially because the essay by Lindbergh continued to tell us that aviation, using it symbolically as well as for its own right, brings two great dangers. One, peculiar to our modern civilization, the other older than history. Since aviation is dependent on the intricate organization of life and industry, it carries with it the environmental danger of people too far separated from the soil and from the sea. The danger that physical decline, which also goes with the high intellectual development, of that spiritual decline, which seems inevitably to accompany an industrial life, of that racial decline, which follows the physical and spiritual mediocrity. So am I removed so much from the soil and the sea that I can only connect to a piece of plastic? Is he right? I would really hate to prove him right. This piece of plastic was really the only thing I could connect with. And then it reminded me actually that we can do something with it. And we can talk about the agency of those new materials, how they're affecting life, how they can become an active substrate. It reminded me of a paper that you must have read back in 2009. It talks about the fact that a material, the consistency of a material, in this case it's a PDMS, which is a form of a silicon, a very clear silicon, which again, it's of that lineage of those biomaterials that we were talking about, can actually affect the fate of differentiation of embryonic stem cells. So if that material is very soft and jelly-like, the cells would become fat cells. If this material is hard and brittle, those cells would become bone cells. And in a sense, the information within the cells have very little to do with the fate that those cells are taking. The substrate is actually the important thing. The DNA, this database, this fetishized genome that we've seen before, is only there to interpret what's going on outside, but it's not the thing that really determines the fate. It's actually what's happening outside as opposed to what's happening inside. And we're now developing a series of prototypes around this work, or around this concept with the work which is called crude matter, that we shown for the very first time in Poland last year, or actually earlier this year, and we're just about to show another version of it in Maribor in Slovenia in about 2 weeks. And just to finish, this type of object that we decided to make, which kind of deals with an abstraction of the golem, because it also deals with kind of the idea of the golem to a large extent. When we showed it in Gdansk in Poland, we then went outside, and there was one of those ancient totems, which were like one of the very first things that the pagan pre-Polish people were worshipping, that just looked the same, which was a very strange coincidence. And I'll leave it there. Thank you.